Namaste, this is Srinivas sir. You are watching Untold Botany. Now I am going to cover the topic nucleic acids. Yes, all of we are very familiar with the nucleic acids. Nucleic acids were discovered by Frederick Mischer, and the term nucleic acids was coined by Altman. Frederick Mischer discovered the nucleic acids and named them as nucleins, but the term nucleic acid was coined by Altman. Where they are present, in the nucleus they are present. On that basis itself, the name is given nucleic acid. Okay, how many types of nucleic acids are there? There are two types of nucleic acids. What are they? DNA, RNA. Deoxyribonucleic acid and ribonucleic acid. So what is the function of the nucleic acids? DNA acts as genetic material. RNA has many functions like catalytic function, as in the case of ribozyme. It can transfer amino acids. It contains that information for the synthesis of the protein, so messenger RNA. Even RNA is structural component of the ribosomes. Like that, many functions are there. DNA acts as genetic material. Even RNA can also act as genetic material in the case of viruses. Here, nucleic acids, both nucleic acids are present in all the living organisms except virus. In virus, either RNA present or DNA present, but no virus contains both the DNA and RNA. For example, if you take the phytophages that attack on the plants, so most of them they have RNA as their genetic material. When they have RNA as their genetic material, then no DNA. Actually, DNA is a genetic material, but some viruses they have only RNA. In such cases, RNA itself would act as genetic material. What does it mean? In addition to the DNA, RNA can also act as genetic material in the case of viruses. So the genetic nature of the RNA was revealed by Frankel Conrad while working with TMV. Like that, nucleic acids they have different functions. RNA which is present in the viruses is called genetical RNA, whereas the RNA that is present in all other prokaryotes and eukaryotes is called non-genetical RNA. rRNA, tRNA, mRNA. These are non-genetical RNAs, whereas the RNA which is present in the viruses is genetical RNA. Okay, that is another topic, leave it. Coming to the nucleic acids, DNA and RNA. How these names are derived? Deoxyribonucleic acid, ribonucleic acid. What is the basis for the nomenclature of the DNA and RNA? Actually, the names are derived on the basis of the sugar molecule that is present in the nucleic acids. Actually, nucleic acids they have pentose sugars, 5 carbon sugars. It may be deoxyribose sugar or ribose sugar. Here, ribose sugar is present in RNA. Deoxyribose sugar is present in DNA. If ribose sugar is present, such nucleic acid is called as RNA, ribonucleic acid. If deoxyribose sugar is present, such nucleic acid is called DNA, deoxyribonucleic acid. So here, the nucleic acids DNA and RNA, their names are derived on the basis of the sugar molecule present in them. So this is one aspect. Coming to this DNA or RNA, these nucleic acids, are they negatively charged or positively charged is the question. They are negatively charged. Why? What is the reason for the negative charge of nucleic acids? Here, nucleic acids, in addition to this sugar molecule, they also have phosphates. So phosphate is present in the form of H3PO4. Yeah, you can see here H3PO4. This can donate a proton. It is donating proton means acting as acid. So this proton donor is present in nucleic acids, RNA or DNA. Hence RNA, DNA also they acquire acidic nature. By donating the protons, it acquires negative charge. So DNA, RNA also acquire negative charge. So nucleic acids are negatively charged because of the presence of the phosphate in the form of phosphoric acid in the nucleic acids. And they are acids. The acidic nature of the nucleic acids is because of again phosphate itself. Because phosphates can donate protons. This proton donor is present in DNA and RNA. So due to the presence of phosphate in the form of phosphoric acid, DNA, RNA, they acquire acidic nature as well as negative charge. So negative charge is because of phosphate and acidic nature also because of phosphate. And the names are derived on the basis of the type of sugar molecule present in the nucleic acid. Okay. Coming to the composition of the nucleic acids. Which chemicals are present in the nucleic acids? What are the components of the nucleic acids? See, nucleic acids, they have nitrogen bases, sugars, phosphate in the form of phosphoric acid. Nitrogen bases are basically two types, purines, pyrimidines. Adenine and guanine are called purines, whereas the cytosine, thymine are called pyrimidines. In the nature, 5 types of nitrogen bases are there: adenine, guanine, cytosine, thymine, uracil. But each nucleic acid consists of 4 types. For instance, if you take the DNA, DNA consists of adenine, guanine, cytosine, thymine. Like if you take RNA, RNA consists of adenine, guanine, cytosine, uracil. Each nucleic acid consists of 4. What is the nitrogen base which is present in RNA, absent in DNA? That is uracil. And the nitrogen base which is present in DNA, absent in RNA, that is thymine. And the nitrogen bases that are common to both DNA and RNA, there are 3. What are they? Adenine, guanine, cytosine. Adenine, guanine, cytosine are present in both DNA as well as RNA, whereas the thymine is present in only DNA and uracil is present in only RNA. This is the distribution of the nitrogen bases. Okay, coming to the purines. Adenine, guanine, purines. Pyrimidines, cytosine, thymine, even that uracil also. What is the basic difference between the purines and pyrimidines? The basic difference is purines have 2 rings, 2 carbon nitrogen rings, whereas the pyrimidines have 1 carbon nitrogen ring. So dicyclic rings are called purines, whereas the monocyclic or unicyclic rings are called as pyrimidines. How many purines are there? 2. What are they? Adenine, guanine. How many pyrimidines are there? 3. What are they? Cytosine, thymine, even uracil. In the case of RNA. In the case of DNA, 2. So purines and pyrimidines ratio 2:3 in the nature, but in each nucleic acid, purines and pyrimidines ratio 1:1 itself. 2 types of purines and 2 types of pyrimidines. If you take the RNA, adenine, guanine, purines, cytosine, uracil, pyrimidines. If you take DNA, adenine, guanine, purine, and cytosine and thymine, pyrimidines. Like that. Okay, coming to the nitrogen bases, purines and pyrimidines are there. If you see the structure of the purines, so here the purines are here, adenine, guanine. Purines are the 9-membered rings. 1, 2, 3, 4, 5, 6, 7, 8, 9. Even here also 1, 2, 3, 4, 5, 6, 7, 8, 9. So purines are the 9-membered rings, whereas the pyrimidines are the 6-membered rings. 1, 2, 3, 4, 5, 6, like 1, 2, 3, 4, 5, 6, 1, 1, 2, 3, 4, 5, 6. So pyrimidines are the 6-membered rings, purines are the 9-membered rings. Okay, they contain nitrogens, that is the nitrogen bases. So what are the positions of nitrogens, where the nitrogens are present? Nitrogens occupy 1, 3, 7, 9. 1, 3, 7, 9 positions. Nitrogens are present in the case of purines. In the case of pyrimidines, nitrogens are present on first position and third position, 1 and 3rd. 6-membered rings and nitrogen present on 1 and 3rd positions. Here 9-membered rings and nitrogen present on 1st, 3rd, 7th, 9th positions. Like this, 2 types of purines are there. Even here, pyrimidines. 2 purines, 3 pyrimidines. Coming to the structure of the sugar, here another component, nitrogen bases and sugars. Which type of sugars are present in the nucleic acids? Pentose sugars are present. What are the pentose sugars? 5-carbon sugars are called pentose sugars. So how many types of pentose sugars are there? 2 types of pentose sugars are there. What are they? Ribose sugar and deoxyribose sugar. So ribose sugar, you can see here, C5H10O5, whereas deoxyribose sugar, C5H10O4. By watching the formula itself, we can know the difference. Here, in the case of ribose sugar, 5 oxygen atoms are present, whereas in the case of deoxyribose sugar, only 4 oxygen atoms are present. Here 5 in the ribose sugar, here 4. 1 oxygen is less, that is why it is called deoxyribose. And here this is called ribose. Ribose sugar consists of 5 oxygen atoms, whereas the deoxyribose sugar consists of 4 oxygen atoms. And if you see here, this is the first carbon, second, third, fourth, fifth. Here I said 1 oxygen is deficient in the DNA. Where? At which carbon oxygen is deficient? Here you see the deoxyribose sugar on the second carbon, two H's are present. But in the case of ribose sugar, on the second carbon, OH present. So here a free hydroxyl group is present in the case of ribose sugar, whereas in the case of deoxyribose sugar, instead of the free hydroxyl group, only hydrogen is present. That is the reason for the stability of the deoxyribose sugar. So here only hydrogen, whereas in the case of ribose sugar, it consists of free hydroxyl group on the second carbon, so that it is chemically highly reactive, so that low stable. So here unstable, this is stable. That is why ribose sugar containing RNA is unstable chemically, whereas the deoxyribose sugar containing DNA is stable, because in the deoxyribose sugar, instead of the free hydroxyl group, only hydrogen present, whereas in the case of ribose sugar, here free hydroxyl group is present. Like this, 2 types of sugars are present in the nucleic acids. Coming to the third component, that is nothing but phosphate. Phosphates are present in the form of phosphoric acids. So here, this is the phosphate, H3PO4. Here it can donate the proton. If you donate proton, it acquires. What does it acquire? It acquires negative charge. So it can donate the proton, so that DNA and RNA are acids, and by donating the proton it acquires negative charge. That is why the nucleic acids are negatively charged. Like this, in the DNA and RNA, in the nucleic acids, all the three types of components are present. Okay, here these individual components, they unite, resulting in the formation of two types of structures known as nucleosides and nucleotides. So what are the nucleosides and what are the nucleotides? Here, nucleosides have two components. What are they? Sugar plus nitrogen base together make up nucleoside, whereas the sugar, nitrogen base and phosphate all together make up nucleotide. You see the nucleosides, you take a sugar molecule plus adenine, nitrogen base, sugar and adenine, that results in the formation of adenosine. Adenosine is a nucleoside of adenine. Like that, sugar, cytosine, that results in the formation of cytidine. Cytidine is the nucleoside of cytosine. Like that, sugar plus guanine. So this guanine and sugar, they together make up guanosine. Guanosine is a nucleoside of the guanine. Like that, here you take the sugar molecule plus thymine. They make up thymidine, that is a nucleoside. Like that, sugar molecule and uracil together make up uridine. So these are the nucleosides. Nucleosides consist of two components, nitrogen bases and sugar molecule. Here, how many types of nucleosides are possible in the nature is the question. Here clearly I have written 5 types are there, but actually 8 types are present. How we get 8 types? If you take the uridine, present in only RNA. If you take the thymidine, present in only DNA. What about this remaining adenosine, guanosine and cytidine? Actually adenine, guanine, cytosine, they are common to both DNA and RNA. Na? So these are common, but sugar molecules are different. Na? If it is ribose sugar, that will be riboadenosine, riboguanosine, ribocytidine. And however, thymidine is present in DNA, not in the RNA. So RNA consists of 4 types of the nucleosides. What are they? Riboadenosine, riboguanosine, ribocytidine and uridine. Whereas the DNA consists of 4 types of nucleosides. What are they? Deoxyriboadenosine, deoxyriboguanosine, deoxyribocytidine and thymidine. So here, depending on the sugar molecule, here 8 types of nucleosides are possible. However, uridine present in RNA, thymidine present in DNA, but the remaining are there. Na? Remaining are common to both DNA and RNA. Na? So depending on the type of sugar molecule, it may form riboadenosine or deoxyriboadenosine. Like that, riboguanosine, deoxyriboguanosine. Like that, ribocytidine or deoxyribocytidine. So ribo 3, deoxyribo 3. 3 plus 3, 6 plus thymidine 1 plus uridine 1. So totally 8 types of nucleosides are possible. So nucleosides, they consist of only sugar and nitrogen base. Here always the nitrogen base is added at the first carbon of the sugar, pentose sugar. It may be ribose or deoxyribose. So coming to the nucleotides, what are the nucleotides? Nucleotides consist of sugar, nitrogen base and phosphate. Sugar and nitrogen base together constitute nucleoside. Na? So here directly we take nucleoside, adenosine, a nucleoside. If you add phosphate, so phosphate is added to the adenosine, resulting in the formation of nucleotide. That is nothing but this AMP, adenosine monophosphate. Here adenosine means adenine plus sugar plus phosphate also there, adenosine monophosphate, AMP. Otherwise adenylate, adenylate, adenylate acid, or this adenosine monophosphate, that is a nucleotide. Like that, guanosine plus phosphate, guanosine monophosphate. Otherwise, guanylic acid or guanylate. Like that, cytidine nucleoside plus phosphate. That results in the formation of cytidine monophosphate. So that is nothing but cytidilic acid or cytidylate. Like that, thymidine plus phosphate. Thymidilic acid or thymidylate. Uridine plus phosphate. Uridylic acid or uridylate. So uridine monophosphate, thymidine monophosphate, and cytidine monophosphate, guanosine monophosphate, adenosine monophosphate. These are the nucleotides. Nucleotides contain sugar, nitrogen base, phosphate. Whereas the nucleotide contain sugar, nitrogen base, phosphate. Whereas the nucleoside contains only two, sugar, nitrogen base. In between the sugar and nitrogen base, glycosidic bond is present, and in between the sugar and phosphate, ester bond is present. Like this, here also 8 types of nucleotides are possible. How? So if you take ribosugar, ribonucleotides, 3. If you take deoxyribosugar, deoxyribonucleotides, 3. 3 plus 3, 6. However, one uridylate present in the RNA and thymidylate present in the DNA. Like that, 8 types of nucleotides are possible and 8 types of nucleosides are possible. These nucleotides, they make up a polynucleotide strand, which will be discussed in the next class.